Hello students, I am Courtney, a student medical doctor at the University of Yaoundé 1. We are going to be continuing our lecture on cytology. Now in this part of the lecture, we are going to be talking about the cytoplasm, the cell organelles and the nucleus. Now let's dive into business. The cytoplasm, that is the cell forming material. It is the cellular material that is found between the plasma membrane and the nuclear membrane of the nucleus. This therefore means that the nucleoplasm, which is found within the nucleus, is not part of the cytoplasm. The cytoplasm can be divided into three parts. You have the cytosol, the organelles and inclusions. The cytosol is the fluid component of the cytoplasm. Now it is made up of a complex mixture of molecules which include protons, salts, sugars and other variety of molecules. Now the organelles within the cytoplasm can be divided into two. You have non-membranous bound organelles. That is those organelles that are not actually surrounded by a membrane, that is the cytoskeleton, centroids, ribosomes. And you have membrane bound organelles that are actually surrounded by a membrane. Now these membrane bound organelles can further be broken down into two. You have double membrane bound organelles and single membrane bound organelles, as we are going to see. Now there's a difference between the plasma membrane and the membrane that encloses our organelles. This difference is due to the absence of the glycocalyx on the membrane that actually surrounds our organelles, that is actually present on the plasma membrane. Now, why is it so important to have membrane bound organelles? Now it must be clearly noted that some organelle content are very harmful to the cell itself. So this organelle content must be contained and must be kept from having contact with the cytoplasm of the cell. For example, you have lysosomes which have hydrolytic content that can be very, very dangerous to the cell. The mitochondria. The mitochondria is a thread-like organelle that is known by many scientists as the power plant of the cell because of its major role in production of ATP. Now, the metabolic activity of a cell can be noted by the density of the mitochondria because of its energy producing role. That is cells that are highly metabolically active will have a high density of the mitochondria, and cells that are poorly metabolically active have a low quantity of the mitochondria. Now it must be clearly noted that the mitochondria is a doubled membrane bound organelle, that is an inner membrane folded into crystals that surrounds the matrix of the mitochondria, and an outer membrane that is separated from the inner membrane by an intermembrane space. Now it must also be clearly noted that the Krebs cycle actually occurs in the matrix of the mitochondria, and the electron transport chain actually occurs on the crystal of the inner membrane of the mitochondria. This therefore brings us to a conclusion that aerobic cellular respiration occurs in the mitochondria. Now anaerobic cellular respiration, that is glycolysis, occurs in the cytosol of the cytoplasm. Now it must be noted also that the mitochondria has its own circular DNA, its own RNA, and it's capable of self-reproduction. Ribosomes. Ribosomes are non-membrane-bound organelles that are formed from basically two components, that is proteins and ribosomal RNA, all derived from the nucleolus of the nucleus. These different components, that is the protein and the ribosomal RNA, are all assembled to form the ribosome in the cytoplasm of the cell. Now, ribosomes are basically made up of two globular subunits, that is the larger subunit and the smaller subunit. Ribosomes are the site for protein synthesis. Now, ribosomes can be divided into two. You have ribosomes that freely float in the cytoplasm, that is free ribosomes, and ribosomes that are found bound to the endoplasmic reticulum, that is the rough endoplasmic reticulum. Now, it must be noted that free ribosomes are actually involved in formation of proteins for the cytosol, while ribosomes bound to the rough endoplasmic reticulum are actually involved in formation of proteins that will be exported out of the cell and proteins for the plasma membrane. Now, we go into the endoplasmic reticulum. Now, the endoplasmic reticulum is an extensive system of interconnected tubules and parallel membranes enclosing a fluid-filled cavity called the cistern. Now, the endoplasmic reticulum is also a double membrane-bound organelle. Now, the endoplasmic reticulum, its membrane, specifically the rough endoplasmic reticulum, is continuous with the nuclear membrane of the nucleus. Now, based on the presence or the absence of ribosomes bound to the endoplasmic reticulum, the endoplasmic reticulum can be divided into two. That is rough endoplasmic reticulum, that is those that contain ribosomes, and smooth endoplasmic reticulum, those that lack ribosomes. Now, these different types of endoplasmic reticulum have their different functions. Now, the rough endoplasmic reticulum is actually involved in folding of proteins formed from the ribosomes that are found bound to them. Now, these folded proteins are actually enclosed into vesicles and are exported to the Golgi apparatus, where it is the center of distribution. The smooth endoplasmic reticulum. The smooth endoplasmic reticulum, as we have said, actually lacks ribosomes, so therefore have no role in protein synthesis. Now, the smooth endoplasmic reticulum has basically four functions, as we are going to see. That is, they are involved in lipid metabolism, synthesis of steroid hormones. Example, you have testosterone, estrogen, absorption and synthesis of fats, detoxification of drugs, pesticides, carcinogens. They are basically and highly concentrated in the liver and the kidneys. Now, the Golgi apparatus is a very important organelle. The Golgi apparatus is a very important organelle due to the fact that they act as distribution centers. Now, they act as traffic directors of proteins formed from protein synthesis. Now, they are actually the Golgi apparatus are actually connected to the rough endoplasmic reticulum by a convex face or a cis face, and they are actually connected to the plasma membrane by what? The trans or the concave face. Now, The Golgi apparatus has basically three functions. Like what that is, it's actually its distribution function. That is modification of proteins. Modification of proteins, that is the addition of carbohydrates. The addition of carbohydrates to proteins to form glycoproteins in a process known as glycosylation. Packaging, that is enclosing them into vesicles, be it for the lysosome, and you have sorting, that is the arrangement of the different molecules that are to be exported to the plasma membrane or to be secreted out of the cell. So we see that from its modification function, its packaging function, its sorting function, they all describe the role of a distribution center. This all explains what we have said. Now, lysosomes. Lysosomes are known as the suicide back of the cell. Now, lysosomes are a single membrane bound organelle. Which contain hydrolytic enzymes, specifically acid hydrolysis. Why? Because their content has an acidic pH and they contain hydrolytic enzymes. So this therefore gives us the name acid hydrolysis. Now, because of their hydrolytic content, they are mainly found in phagocytic cells, that is macrophages, neutrophils, and other cells that are involved in basically immune activity. Now, lysosomes basically have three functions, that is digestion. As you can see, digestion. Now, it must be noted that during endocytosis, that is bulk transport of materials into the cell, which we can have two types. Phagocytosis, if the transport content is particulate in nature, and pinocytosis, if the transport content is solid in nature. Now they help. Now within these endocytic vesicles during endocytosis, we have important nutrients like glycogen, other large molecules that have to be broken down into smaller molecules using lysosome. Now you also have the removal of worn out tissues. Now removal of worn out tissues or organelles. Now it must be clearly noted that organelles have their expiring dates. That means there are dates beyond which they can no longer become functional. So these organelles must be digested so that the building blocks that were used to form these organelles can be recycled to form another organelle. Now this is seen during autophagy, that is self-eating. Now there's also self-destruction seen in conditions of apoptosis, where the cell undergoes destruction, that is self-digestion, that is autolysis. Now this all explains further functions of the mitochondria, which are classified under basically the three functions, that is digestion, removal of worn out tissue, self-destruction. Now peroxisomes. Peroxisomes are also membrane-bound organelles that actually contain two enzymes, that is oxidase and catalase. Now it must be noted that in the absence of oxygen, the mitochondria begins to produce reactive oxygen species that is free radicals that have a very free radicals are very, very reactive species that react with almost all biological molecules within the body, and it's very dangerous. Now let's see the role of oxidase and catalase to counter the effect of formation of free radicals. Now, oxidase helps to detoxify these free radicals formed during low oxygen level. Now, but another problem comes up. In the course of detoxifying the free radicals leads to the formation of another dangerous molecule, that is hydrogen peroxide. But now, how do peroxisomes counteract this effect? With the use of catalase. Catalase detoxify hydrogen peroxide or breaks down hydrogen peroxide to water and oxygen. Now, it must be clearly noted that unlike lysosomes that are formed from the Golgi apparatus, peroxisomes are not formed from the Golgi apparatus. The cytoskeleton. The cytoskeleton is a very, very important non-membranous organelle because they are actually involved in keeping the architecture or the 3D dimensional structure of the cell. Now they are basically divided into three types. You have microtubules, microfilaments, you have microtubules, which you find them in centroids, you find them in flagella, you find them in cilia. You have microfilaments. Example, you have actin, found in myofilaments of muscles. You have intermediate filaments. Now, the major function of the cytoskeleton, like we have said, is to maintain the architecture of the cell. They also help in cell movement, like the movement of cell organelles. The movement of cell organelles within the cell has seen during cell division. Now, this is an assignment to be able to describe the different portions of the cytoskeleton, describe the cellular extensions, distinguishing between them, give examples of their location and function in the human body. Okay, students, but what I want us to remember importantly concerning microtubules is the arrangement in cilia, flagella, and centroids. Now, it must be clearly noted that in centroids we have 9 triplets of what? We have 9 triplets of microtubules in centroids. Now, in flagella and cilia, we have a single pair of unfused microtubules surrounded by 9 duplets of microtubules. Now we are going to go to the next part, that is the nucleus specifically. Now, the nucleus is known by many scientists as the control part of the cell. Is known by many scientists as the control house of the cell because of the presence of the genetic material that controls protein synthesis. Now, the nucleus is it must be noted that not all cells are nucleated. Some cells are anucleated. For example, we have platelets, we have red blood cells, specifically mature red blood cells. Also, we have cells that are mononucleated and multinucleated. For example, that is mononucleated, that is one nucleus. Like for example, hepatocytes, macrophages, and we also have cells that are multinucleated. For example, skeletal muscle cells. Osteoclast, that is bone destruction cells, that is the osteoclast. Now, the nucleus is divided basically into three compartments, that is the nuclear envelope, the nucleoli, and the chromatin. The nuclear envelope, the nuclear envelope, that is the nuclear membrane. The nuclear membrane is a double bounded membrane. That is, the nucleus is a double membrane bound organelle. Now the membrane is divided into an outer membrane that is continuous with a rough endoplasmic reticulum, and an inner membrane made up of lamina that is involved in maintaining the shape of the nucleus. The nuclear membrane actually surrounds a fluid-filled cavity, that is the nucleoplasm, which is similar to the cytosol, containing proteins and other molecules. Now, the nuclear envelope, there's a connection between the nucleoplasm and the cytoplasm. That is, the nuclear envelope contains nuclear pores that allows the connection between the nucleoplasm and the cytoplasm. This is very, very important when concerning the transfer of the messenger RNA from the nucleoplasm to the cytoplasm. The nucleoli. The nucleoli is a dark staining spherical body found within the nucleolus. Now, it is not membrane bound. Now, it must be clearly noted that this is the site of the production of the components of ribosomes, that is the protein part and the ribosomal RNA. But it must be also noted, like the way we said once, that ribosomes are assembled in the cytoplasm. Chromatin. Now, before we talk about chromatin, it must be clearly noted that our genetic material is long as almost 1.8 meter, but it is able to fit into a cell with diameter of nanometers. How is this possible? Now, it is due to the presence of a folding of our genetic material, that is the DNA, around a globular protein called histones. So the chromatin is composed of 30% of the genetic materials, that is the DNA, 60% of globular proteins known as histones, and 10% of RNA. Now, it must be clearly noted that the chromatin can be divided into two. The chromatin can be divided into two. That is the euchromatin. The euchromatin that contains what? The euchromatin that is less condensed and opened for transcription, and the heterochromatin that is more condensed and closed for transcription. That is, that portion of the chromatin is not available for transcription. Now, the interaction between like we said from the beginning, the interaction between our genetic material and histones enables our very, very, very long genetic material to fit into the nucleus of the cell. Like this explains it all. This explains it all. Now, that is, these are our histones. This is our genetic material. Now, the folding of the genetic material around our histone complex forms what we call the nucleosome. Now, these nucleosomes are linked together by intermediate segments of genetic material. The nucleosome plus these intermediate segments of the genetic material form a bit-like structure. They form a bit-like structure which we called chromatin, fiber. That is, this bead-like structure is what we call the chromatin fiber. Now, if you clearly see from here, the part of your genetic, the part of your genetic material that is actually open for transcription and it is not condensed, that part of your chromatin is what we call the chromatin. Now, this part which is actually condensed and is not available for transcription is actually what we call the heterochromatin. Okay, and this finally brings us to the end of our lecture. Thanks for your kind attention.